What if we could do more than just manage the symptoms of neurodevelopmental disorders? What if we could actually rebuild the brain's own wiring? Today, we're going to dive into a pretty groundbreaking case report that explores a radical idea, using the body's own signals to grow new neurons and, just maybe, reverse the core imbalances we see in autism spectrum disorder. All right, so here's our game plan for today. First, we're going to pinpoint the exact neural imbalance that this therapy is trying to fix. Then we'll unpack the therapy itself before we look at the pretty striking results from 3 clinical cases. We'll visualize how it actually rebuilds brain networks, and then finally, we'll look ahead at the incredible potential this whole approach has for the future. So, first up, what's the fundamental problem in the brain's wiring that contributes to ASD? To really understand the solution, we have to get a crystal clear picture of the problem. To really get a sense of the challenge here, you just have to appreciate the sheer complexity we're dealing with. Look at this. This isn't just a few wires. It's a dense, intricate forest of billions of connections. Now, in a healthy brain, this network just hums along in perfect balance. But in ASD, as you're about to see, even a subtle imbalance can throw the whole system off. And right here, this is the core of the problem. Think of your brain's activity kind of like city traffic. You need go signals. Those come from what we call excitatory neurons to get things moving. But just as important, you need stop signals from inhibitory neurons to prevent total chaos. A really key theory in ASD is that this whole stop and go system is just out of whack. And that leads to disorganized network activity. Okay, so now let's get to the innovative solution that's being proposed in this research. And this isn't some drug to just cover up symptoms. This is a therapy designed to regenerate the very neurons that are needed to bring back that critical balance. The star of the show in this therapy is a group of proteins called cytokines. You can think of them as these highly specific instruction manuals that get delivered right to the brain's stem cells. So the researchers created this precise cocktail of these cytokines to send a really clear message. Hey, we need more stop neurons right here, or build more go neurons over there, all depending on what that specific patient's imbalance is. Now, working right alongside the cytokines are these things called exosomes. These are like little delivery packages, and in this study, they were derived from young porcine brains. They carry this vital cargo, like microRNA that helps support and guide the new neurons as they grow and try to fit into the existing circuits. The whole process is incredibly methodical. First, they use an EEG to figure out the specific nature of the neural imbalance. Then, a custom-tailored cocktail of cytokines and exosomes is given right under the tongue. This mixture kicks off the targeted growth of new neurons, and the ultimate goal, of course, is to rebalance the entire cortical network. So, the big question is, does it work? Let's get to the evidence. We're going to look at 3 really fascinating clinical cases, and all of them were born via in vitro fertilization, which is a key detail we're going to come back to later. So here's a quick look at our 3 cases. And just notice the diversity here. Different ages, different main symptoms, and most importantly, very different underlying brain activity, as you can see from their EEGs. And this highlights a really critical point. ASD isn't just one single condition, and this therapy is tailored to the unique neural signature of each person. Now, this is where it gets really interesting. Case 1's brain was basically too loud. It was hyperexcitable. But in contrast, Case 2's brain signals were too quiet. They were underdeveloped. These are essentially 2 sides of the same coin, the coin of imbalance, and they each required a totally different regenerative strategy.
And the results here are just visually striking. Okay, focus on the left image first, for Case 1. See those tall, spiky red waves before treatment? That's the brain being too loud, right? Hyperexcitable. Now look at the black lines that were recorded after the treatment. So much calmer, so much more regulated. This suggests that therapy successfully grew the inhibitory stop neurons that were needed. Now, shift over to the right for Case 2. It's the complete opposite story. The first red signal is weak, almost flat. But after treatment, look at that robust, healthy black wave. In this case, the therapy successfully built up the excitatory go signals.
For Case 3, the big transformation was in emotional processing. Just look at the graphs on the left, before any treatment. The brain's electrical response to different emotions is just a messy, weak jumble. Now look to the right. After the therapy, you see these clear, powerful, distinct peaks for joy, sadness, and anger. The brain has essentially relearned how to properly tell the difference and process these really fundamental feelings. So we've seen the before and after data, But how exactly is this therapy rebuilding these incredibly complex networks? Let's take a closer look at the actual mechanism. This little schematic shows us exactly how it works at the circuit level. The big cells are the excitatory go signals, but look at that intricate web of smaller cells all around them. Those are different kinds of inhibitory interneurons, and they form the brain's really sophisticated braking system. Each type has a very specific job, like controlling the timing or the strength of the main signal. The therapy works by precisely regenerating the specific brake cells that were missing or underdeveloped, and that brings the entire circuit back into balance. What's truly revolutionary here is the idea behind it. The researchers believe this therapy is basically guiding a process that mirrors the brain's own natural development. It's not about just adding some synthetic chemical to mask a problem. It's about rebuilding a complex, functional network using the brain's own fundamental building blocks. So this brings us to the bigger picture. Why might these imbalances have happened in the first place? And what could this technology mean for the future of treating brain disorders? A really critical thread running through this whole report is that all 3 patients were conceived using IVF. So the researchers had to ask, is there some kind of connection here? The paper proposes a potential link through something called epigenetics. See, processes that are part of IVF, like hormonal hyperovulation or freezing oocytes, they can introduce these epigenetic modifications. Now, these aren't changes to the DNA code itself, but more like changes to how that code is read and expressed. And that could, in turn, alter the very delicate process of brain development.
And this idea connects to a much, much wider field of research. IVF is just one of many prenatal environmental factors. I'm talking from maternal nutrition and stress to chemical exposures that can influence a fetus's epigenetics and potentially shift the course of neurodevelopment.
This brings us to the most exciting possibilities. Because this therapy targets such a fundamental mechanism, that stop-and-go balance in the brain, its potential isn't limited to just this one specific form of ASD. The authors are suggesting it could form the basis for treating other neurodevelopmental disorders. It might even offer some hope for neurodegenerative diseases like Alzheimer's, where the goal would be to regenerate the specific neural circuits that have been lost over time. And this leads us to the final powerful question. Look, while this is just a report on 3 cases, it represents a potential paradigm shift. It asks us to start thinking about a future where we don't just manage neurological conditions, but we actively repair and regenerate the brain. It's a really tantalizing glimpse into what could be the next great frontier of medicine.